I think we will start with a round of presentations. I'm Gustavo Dziewczapolski, Scientific Director of CMD, and also a foundation building strength for nemaline myopathy. And
Oh, sorry.
I'm a PhD completing my dissertation there with Dr. Kristen Baum. I also have ultra rare form of limb-girdle muscular dystrophy, specifically the Fukutin subtype. So, yeah.
I'm Chiara Manzini. I'm an associate professor a few miles over there at Rutgers in New Jersey. And I've spent a long time working on the genetics of these retinopathies, looking at several of the genes. And now we've been developing several zebrafish models, and I can explain why zebrafish instead of mouse, and basically trying to figure out what is going on with these diseases that affect multiple parts of the body.
And I'm Kathy Mathews. I'm at the University of Iowa and have for 20 years run a natural history study of dystroglycanopathies across subtypes.
Hi, I'm Trish Laria. My son is 26. He has limb-girdle. He's, what did I say? What was? TRAPC. TRAPC is the gene, and he is really affected. He has seizures, intellectual disability. Right now, he's mostly wheelchair and bedbound. And the disease is definitely getting worse.
I'm Dima Sadi. I'm a pediatric neurologist and neuromuscular specialist. I work with Dr. Mathews at the University of Iowa.
My name is Charlie Brown. I'm the grandpa of Nova. Bill Brown, my daughter is Nova. She has one of the dystroglycanopathies, ultra rare one as well, INPP5K. Dr. Manzini is one of the people who discovered it. I believe you're on one of the papers, which is really exciting to be here for that. So, yeah. And we've also been to the Iowa thing. I recognize Dr. Mathews, so kind of cool.
okay, well, when the other family comes, we will ask them to introduce themselves. But okay, so this is the research session. So the idea is to give you some brief description of we are doing in the labs, and of course your questions, and discuss whatever you think you want to discuss about the part of the research of the disease. We only, well, doctors and seeing patients, right? But the other two, you don't see patients. Okay, so I don't know, you want to start with your research, explaining the why zebrafish, and what genes are you studying, and how can we learn, because we are not working on
So I started with the genetics because there's 2 of you here, but if there were 10 of you here, most people would have a different, each person would have a different genetic cause. Maybe do we want to?
Just start.
We just started, so let's finish things. My husband is finishing up with my son. He'll be here.
Okay, you want to introduce yourself?
Wow, I need the
We don't buy it.
It's a very small group, I see.
Yes.
Do I really need this? Okay, my son Tyler has a POMT1 mutation. He's 3.5, and my husband Zach will be joining. We're from Long Island. He was diagnosed at 15 months. I'm not sure what else you want me to. And we're being seen at CHOP now. We switched recently from our hospital on Long Island.
We just started to talk about what people is doing in the lab about research on these diseases.
That's why one of us would have just normally taken my son out, but we both really want to be here to hear this. So excited to hear.
So, I'll introduce myself very quickly. Chiara Manzini, Rutgers School of Medicine. As I was saying, when I started in this field, which was 20 years ago, we were really just trying to find the mutations. There were 6 genes known at the time. Now, depending on who you ask, there are 18, 19, 20, depending on, depending on the presentation. But what happens basically is that there is this protein, dystroglycan, that sits on the surface of the cells that interacts with a lot of other proteins in different tissues. And it does this through a very specialized chain of sugars that is assembled in a very specific order for this interaction, which was in large part characterized by Dr. Campbell, who's at Iowa, that works closely with Dr. Mathews. Every single enzyme that builds this chain can cause a form of dystroglycanopathy. So when I started, we just kind of like had the beginning, and then we did more and more and more genetic research because a small subset of patients could receive a genetic diagnosis, and so basically the doctors were going to be able to tell you what gene was mutated in the child. And so I was involved in discovering some of these genes, in looking around the world at different populations in different countries and figuring out which genes were mutated there. And right now we are finishing a large study trying to get as many patients as we can with the most severe forms of dystroglycanopathy, which actually tend to be lethal in like the first few years of life, and really trying to figure out if we can say which genes are mutated in those. And so in my talk on Monday, I will talk about how the beginning of that sugar chain and the genes that make the beginning of that sugar chain, so POMT1 being the very first one, are the ones that are more correlated with having more severe disease, while the ones later in the chain we don't find quite as much in our groups of patients. And because this is so rare, it took a very long time to get enough with a very defined presentation just to be able to say that. This said, because there are so many genes and every single one of those enzymes attaches a specific sugar, what we started doing with the zebrafish is to make as many mutants in succession as we can. Right now we have 4, and then the issue is like we're going to be to continue, and we're working with people that have other mutants, and kind of like everybody trying to get together to see how many of these can we compare exactly in the same way to get at some of the questions that Gustavo was talking about, like, are they different? Can we figure something out that could help understand the disease better? So right now we've published POMT1, we have POMGNT2, POMGNT1, IMPP5K, because we've made the IMPP5K, and then I'm talking to people who have B4GAT1 and all the DPMs and several other mutations. And one thing that we found that was very interesting, that we're really trying to understand, is that POMT1 seems to be different, even though the muscle disease looks the same, has the enzyme that comes right after it. And if you want, then with the questions I can explain in more detail. we also see an impact in POMT1 on the neurons that control muscle function. So it's not just muscle disease, but we also, and this is in the fish. And so we're really interested in trying to figure out if that's also the case in humans, because that completely changes how we address the disease.
Can I ask a question?
Sure, of course.
Interactive?
Yeah, yeah.
So when you say that I'm sorry. Oh, I was wondering why that we needed a microphone in such a small space. Okay, thank you. When you say that the neurons that control muscle function, you're seeing a difference in the POMT1 with the zebrafish. What do you mean by the neurons that control muscle function? Like, what do you see that's different? Sorry.
Yeah, I think we have to.
So we know that POMT1 controls the development of the brain, right? That is already known, but what we're seeing is also a loss of the very ends of the motor neurons that come from the spinal cord, and
To orient you to what she's saying. So if I want to move my right hand, the message starts at the head. It goes from my motor cortex down my spinal cord to a cell in the spinal cord. And that is the motor neuron. That's the cell that's sick in spinal muscular atrophy, if you've heard of that disease. And then that cell sends the message, which is along a nerve, down to the muscle, which is what is primarily affected in muscular dystrophies, and the muscle is activated, which allows the limb to move. So she's saying that we think of POMT1 and the rest of the diseases here as being primarily muscle diseases, but we also know that the dystroglycanopathies can affect retina and brain, because that dystrophin glycoprotein complex is also found in those tissues, and that the anterior horn cell, which is the cell in the spinal cord, might be also infected. To orient you.
affected, yeah. So there are neurons throughout your back, and so the neurons that control my leg are actually at the bottom of my back, and the brain is sending them messages, and then there is a neuron at the bottom of my back that sends the message to my leg. The neurons that control my arms are up at the level of my arm, right? So you have cells distributed throughout your entire body that will send that information to move, to contract, and like to basically generate a movement to your muscles. And so we see that the endings of those neurons that come from your, basically from your back, that come all the way to the muscle, are gone. And we're trying to figure out, are they gone from the beginning? Are they not developing properly? Or are they degenerating? And that is important because if there ever was a gene therapy affecting the muscle and that could possibly fix the muscle, if the information is not coming or is not working correctly, that wouldn't work. At the same time, because of spinal muscular atrophy, now there are very good gene therapy for motor neurons, which have been approved by the FDA. And so then we're basically trying to figure out exactly what's going on to see how we're supposed to possibly restore POMT1 function. We do not see the same for some of the other dystroglycanopathy genes, but we're trying to figure out if this is a general problem or whether it's a specific to.
Just to be very clear, the SMA treatment will not work for POMT1, POMT2, or any other dystroglycanopathy. It is specific to the genetic abnormality in SMA. But the concept it's proof of principle that you can fix a missing protein in the anterior horn cells. So I didn't want anybody to go away
Yeah,
with
to
the
think that you need to
misunderstanding.
That you need to take that part. It's just that we know how to get at those cells if we need to get to them. And then you would reintroduce the specific gene, so in this case, POMT1. I think that's it. Then we're working with some maybe 5K fish, and right now we're stuck because we need money. Which is one, I think.
The fish is the only animal model that we have for that gene.
No, so there is a mouse model, and actually, if I saw you, I was going to try and give you the contact of the person in the UK that has the mouse model, because Laura? Yeah. Okay, so that has the mouse model. So basically, those are kind of like the different groups. Yeah.
And for POMT1,
POMT1, so the mouse, Kevin Campbell in Iowa has. The problem with the mouse in POMT1 is that the mouse die embryonically because of a specific difference in how the mouse embryo develops versus the human embryo. So there's been a lot of work done on that mouse, specifically removing POMT1 in different tissues. So there are ways to like genetically just remove a gene in parts of the brain, in the muscle, in different parts of the body. And so a lot of the work that has been done on that mouse has been done with this approach. The fish at this point is the only animal that from conception through development has every single tissue affected, every single tissue where POMT1 is removed. There is a fruit fly, so I'm working with the person who's developed the fruit flies for POMT1, and we're also trying to basically work together to really understand what POMT1 is doing and trying now to figure out if we can make human-derived stem cell and human-derived stem cell models. And I know that Kevin Campbell has a collection of all different kinds of cells, including POMT1, right? Yeah. Oh, Matthew, yeah.
Thank you. So I work on a different subset a bit. So as it was just stated, that growing sugar chain, I focus on sort of 2 steps down from that. So I focus on the CDP ribitol biosynthesis pathway, which has 3 genes that are implicated in that pathway that add on, I believe that's 3 sugars down from like the palm, that area, or more of the start from where alpha-Dystroglycan is. So, but within that, my research actually is not on in vitro studies, but it's actually in silico. So I do artificial intelligence work to basically take all that's known and put it together. And so the idea of my work is to sort of tie everything that's known about the genetic variants and the underpinnings of the disorder, and then correlate that to the actual clinical presentations and the manifestations that are caused, both biologically and then clinically. And then the idea is using AI, we can sort of bridge them together and sort of create networks, if you will, to be able to use different programs to be able for a user to ideally be able to put in any gene of interest, and you would then be able to characterize that disorder, if you will, in both clinical, biological, and genetic even instances. So that's sort of the scope. So in the most broad sense, this approach can actually be used in basically any ultra rare disease. It's pretty cool. So we're focusing on Fukutin, ironically, first, but also not just because I have a unique relationship with that gene, but also because it's very centrally located within this pathway. And so it also has a very well characterized, defined set of disorders that are caused from this gene. And so we're using that gene to sort of model basically other genes as well. And so the idea is we could go to FKRP even, or some of the genes up and down that pathway. And so it's really cool. So I'm happy to do it. So.
Can I ask you, Matthew, what data? So do you have the clinical, all of the clinical information? What do you feed to the AI so that they can understand? Like all of the clinical information? Do you have brain imaging, muscle imaging? Like what do you give it?
So good question. So, I think the mic went. I think we input basically everything that's known available publicly. So basically that would be any sort of imaging findings, anything that any clinician has ever done that's publicly available is being currently fed into these very large models that are being built. It's cool. I can give you guys, we're working with the group at the National Library of Medicine that's characterizing a new tool that harnesses all of these AI models together. It's called Rare Source. So it's actually, it's out available now, but it's still in its infancy. But hopefully all of my work can actually be or will be inputted into this as well. And then hopefully the dystroglycanopathies as a whole are going to be inputted into this work as well. So the idea again would be that you could quite literally search for a gene and then get everything known about that gene and every clinical presentation or every consequence associated with it would also be available to explore, both from the clinician's perspective, but also the patient's perspective and the family's perspective as well. So that's the idea.
And I have a related question about how do you input the data there? You say public information, but so for example, registries like we have in the CMDIR, should we start asking the families that register to consent for the data to be input to an AI system, something like that? Or because, I mean, if it's only in our registry and it's not published, we cannot share the data.
Right. So that's a really good question. So the idea is that right now it's very controlled. So because a very common thing you'll hear when anyone says AI is, the old line is if you give it garbage, it will give you garbage back. And so the idea is we are trying to fine tune the models first internally. And then once we get like really, really good results, then we can start to incorporate data from, all over that may not be publicly available. But that's something that we're actually starting to look into as we start to get closer to a more, external facing version of these tools. So.
Okay. So I would add that as working with the registry, I think it would be a good measure, a good policy to have right now to ask the families to consent for the future, not because they will share the data right now, but in the future, most probably we will need that. So instead of start when it's ready, already have the consent now.
Actually, yeah, and I'll also add, I'm sure you guys have, so there are currently variant repositories, like, the big public ones like ClinVar. For the muscular dystrophies, there's also one called LOVD, which we are pulling from all of these sources. And LOVD and ClinVar are clinician, defined. So it's a little bit more precise. They aren't predictions. It's like this has actually been proven. So the idea is that also I know at NIH with Carson's group. Do you guys know Svetlana Gorkova? Okay, she's on the ClinGen panel. So as ClinGen starts to get more and more fine-tuned, we're hoping that these registries can get fed into those, which then those get inputted into us. So once that sort of medium is there, then yes, the answer is yes. We would be able to extract all of that data to feed it. And the more data you have, I mean, you guys know this probably too, but the better it gets, the more it has. So it's definitely very cool because that was something that we were thinking of too, where, obviously it's evolving. The more families that come forward, the more variants that get uncovered. That's the idea, is that all of that would be available for us to input. So, yeah.
So can I ask another question? Yeah. So because I know now there is a lot of research into doing this in silico clinical trials, like in vitro, basically trying to model how different drugs could change could improve specific disorders. Is one of the goals also to be able to interact with something like that, or?
So I think, so yes, so our group that we work with, that I'm affiliated with too, NCATS, they do a lot of the drug targeting. So they have these mega databases with basically every theoretical, molecular compound that we have, and they'll run these massive screenings on against different genes, and they'll ask the different clinician groups out there or the different researchers, and they'll ask them for that assay, and they'll screen basically if an expert comes to them, they'll basically accept it and do their own findings to help that group explore much deeper into it. So I think the idea for at least our purpose is to more potentially uncover sort of therapeutic entry points, if you will. So not more of the actual targeting aspect, which is like, your second sort of aim. Ours is sort of the preliminary first step, where it's like you actually need to know where you would enter from, and like actually understanding, are we treating the actual condition? Are we going after the gene itself? Are we targeting its downstream effects? Or, like what are we actually sort of trying to target to sort of stop the progression or slow it or anything of that nature? So that's the idea.
The reason I was asking is because the ultra rare, like would be a MPP5K or TRAP, it's because there are so few people, it's really, really difficult to start clinical trials for this. And so there's been a lot of impetus in trying to find ways to at least get to a possible therapeutic that then would make a company or the NIH invested, and they're so different. There were, I don't know, hopefully there will still be all different kinds of like NIH funding, so government funding, requests, requests for application that target that specific, like, ultra rare and development for compounds. Because especially funding contracts, these are going to be some of the first things that may be not considered. And so all of these new tools are really exciting because we can try and find ways to still continue to do research.
And actually, I would like to add to that. The idea that you're saying, really well is actually something that we're exploring as well to add to this, because the idea is a lot of times that, as you said, because they are so rare, it can be very difficult to create a therapeutic, not all the time, but a lot of times, to get the funding to create one specifically for that. So that's why this AI work is very, we think, very important, because the idea is that if we can identify common pathways and things that are common to other disorders that do have therapies, can those therapies also be used to assist? So sort of even starting to enter the realm of drug repurposing, even, or identifying the right cocktail, even. So yeah, that's an endpoint.
So, as far as my son Danny, he is definitely registered. He was diagnosed by University of Iowa. That's where his muscle biopsy went, and he's definitely on the registry. That's all, because that was like 13 years ago. So that's all I need to do for it, because I would do anything to help with the research. But that's all I need to do as a parent, right? Is have him on this. Yeah. Okay. That's all.
Because we were late and we missed the introductions. I'm just curious, because it's such a small group, like you said, your son, do you mind sharing what mutation he has?
The TRAPC.
Okay.
TRAPC 11.
Okay, thank you.
My daughter Nova, 7, has INPP5K.
These two cases in the very rare spectrum, more rare than POMT1. Yeah, and
I'm Fukutin, so it's like the most ultra rare. So it's like almost. What is it? Those ultra rare. Yeah, right.
it's the most prevalent in Japan, right? The Fukutin.
Yeah. The
Limb-girdle form, yes.
the condition, the actual genetic background of it, though, is unique within it. So that's all I'll say.
Okay, so you can mention about part of the research that your center is doing, because
So, I said for about 20 years, maybe 20 to 25. We're still awaiting 13 months and counting, a word from the NIH. A natural history study across the Dystroglycanopathies. So we have, people come for a research visit where we collect patient-reported outcome measures, we do formal physical therapy evaluations, get other information, and hopefully provide information back about the group of disorders or the specific disorder. We collect biologicals, including urine, blood, if the child or family consents, or teenagers often say no. So if the patient consents, and at the first visit, we collect skin fibroblasts. So it was mentioned that we have this biorepository as part of our Wellstone Center. So the Wellstone Center has 3 components. One component is Kevin Campbell's lab, which is to look at, which he's doing basic science of the dystroglycanopathies and has contributed to understanding the pathophysiology and the structure of the molecules. Steve Moore has the biorepository and collects all these tissues that I just mentioned, plus if somebody has a muscle biopsy that was sent to him, a component of that goes to the biorepository, all the skin fibroblasts, so that if a researcher in, Seattle is interested in cells with TRAPC11, I don't know whether we have skin fibroblasts on you, but he could send them a microtome section, so a teeny, tiny section of a muscle with TRAPC. So they would be able to then, it's to facilitate the study. And often, we were talking about therapeutics, often therapeutics might start with a concept or a drug library screening, either through AI or through tissues. Often skin fibroblasts can be used to do part of that testing. What would happen if we put this drug onto or into a cell? What would happen to a cell? So that's a step before animals. And then you can do fruit fly, zebrafish, and then you have to do something furry often, and then I divide between the furry and the non-furry animals. So, and then final is to go to humans. So in terms of the, just in general, a statement about congenital muscular dystrophies versus limb-girdle muscular dystrophies, the distinction between those 2 disorders has varied over the course of the last 100 years, and where you draw that line has varied depending on what paper you talk about. So they, in general, among the dystroglycanopathies, almost all of them are a continuum, even the most severe forms. There are people with FKRP mutations, which is the most common of the dystroglycanopathies, who look just like a POMT1 patient. Most people with FKRP have a limb-girdle presentation and walk into childhood or young adulthood. There are people with POMT1 mutations who, I think POMT1, definitely POMT2, who have more of a limb-girdle presentation. So these are very much spectrum, probably related to the severity of the specific mutations and how much residual glycosylation, how much of those sugar groups remain based on that mutation. So the one other thing I was going to mention is that we also are a clinical trial site across diseases. So we do a bunch of clinical trials, including the 2 open clinical trials for people with Fukutin. They're both limb-girdle, but FKRP, thank you. Got Fukutin on the mind, Matt. FKRP. So one of those, and both of them are pertinent to, I think, the entire dystroglycanopathy population. One is a gene replacement trial, so, many of the dystroglycanopathies seem to be that they would be great for gene therapy because they're often small enzymes, they fit in a virus, you don't need to have, they're not a structural protein, they're not going to turn over. So they are an attractive target for gene therapy. Now, before I go on about that, gene therapy has risks and benefits. In some diseases, it's been a knock your socks off success, but there are risks up to and including death associated with getting the virus that delivers the genes, and there have been several deaths in the neuromuscular community recently. So it's not like a panacea. So people shouldn't be saying, oh, we're just waiting for the gene therapy and then all our problems will be solved, because it's not that straightforward. But with FKRP, there is an active clinical gene therapy trial. So if that shows success, that will be a win for the entire dystroglycanopathy community as, at a minimum, proof of principle. The other mechanism that is currently in clinical trials is what I think of as substrate loading. So these are enzymes, and Chiara mentioned that each of, and I think of this now, I organize this now into enzymes that directly place a sugar onto the growing sugar group. And then there's a group of enzymes that are required to prepare the substrate to place on the growing sugar group. And then, like TRPC11, there are some, like that's a trafficking gene, there are some other mechanisms. So I kind of put them in 3 buckets. FKRP is one that is required to put a sugar onto the growing sugar group. If you give more of the sugar substrate that it uses, we all know that you have to have a little bit of FKRP activity, POMT1 activity. You have to have a little bit of each of these enzymes activity, or you would not grow to be a fetus. You would not be born. So there's a little bit of activity. If you push the residual activity by substrate loading, give them a bunch of whatever it is they act on, can you make it work harder and make more of that growing sugar group? So there's a clinical trial right now in FKRP with that to answer that question. And I don't know the answer to that question. We'll know sometime within the next 1 to 3 years, depending on how an interim analysis goes. If it is effective, then that substrate, in theory, should work for Fukutin and should work for ISPD patients. If it works in FKRP, Fukutin, and ISPD, a different substrate loading mechanism could, in theory, work for some of the other diseases. What?
We're doing that too. We're looking into what substrates.
Yeah. So the kinds of understanding the biology through AI, through animal work, understanding the biology of what the enzyme does and what it works on. If there is effectiveness in one disease, it might work on the others. The concept, the principle could work on the others. There was one other thing. In terms of whenever we're talking about, did you want to show that?
It may be, we were trying to find a picture of where all of the different pieces fit. I have one on my laptop. I have one on my laptop.
I think this one is connected there.
If you zoom into the top. Yeah, it's just that I don't know.
Zoom into the right.
Yeah.
Those are all of them.
Yeah.
So while she's pulling that up, yeah. We also just reported another new gene that is of the more severely affected. When I go back to when we think about therapeutics, the question then comes up, what can we realistically expect from a therapeutic? The eye findings and the brain findings are largely developmental. They are established, in the first trimester of pregnancy. They are determined by the way the role of dystroglycanopathy in helping to create the structure of the brain. There is a question about whether there is also a component of the brain activity that is do you want to come try it?
I didn't want to.
Is it another window? Like it's, like it's a.
But that's what I'm trying to figure out. But I don't want to interrupt too much.
I'd help, but I'm worthless. So there's a question of whether there is also a component of the dystroglycan complex working in the communication between cells in the brain. The structural component of the brain effects that are from the time of conception will not change if you use a therapeutic. If there is an element of the dystroglycan complex being involved in the communication, it is at least in theory possible that you could have some effect on some of the brain manifestations. So I'm not eliminating that as a possibility, but it is less, there is a component that will not change. What people hope for in the congenital muscular dystrophies is that we can improve muscle function by using a therapeutic. Muscle is a dynamic tissue. It turns over, it changes, it grows. It is unlikely that we're going to have a one and done, slam dunk, bingo, fix that problem sort of therapeutic agent. So it is likely that we will require multiple different approaches to managing and improving the muscle component over a long period of time because muscle turns over and changes and grows. So I think it's super important among muscle diseases to make sure that we pursue every possible avenue of therapeutics because we are likely to need more than one to really be a game changer. Another thing that we looked at a few years ago was sort of in the congenital muscular dystrophies where brain function is a big component. We polled parents, and I could poll you guys too, but about, what if you had your magic bullet and you could fix it all, you'd fix it all. Okay, well, let's say that's not possible. You can't fix it all. Is fixing the muscle function important to you? If you could fix the muscle function and not the brain function or improve, we're not going to be able to fix because a muscle that's gone is not going to come back by putting in a sugar. But if you could stabilize the muscle function, how important is that to you? If that was the part, I'm asking here I'm very curious for your input. How important would that be to you? I'll tell you what we heard before was that it would be really important. It would be really important if my child's head control was better. It would be really important if they were less floppy. So I'm just curious about how important is fixing the muscle function to you? Nova's brain is much less involved than some of the dystroglycanopathies.
Yeah, and that's kind of where we are. Nova in particular is, I guess, mild on the spectrum of both brain and muscle. There's definitely some muscle weakness and, growth kind of things, but I don't know what I would choose at this point if I had to fix one because, she is happy, she is social, she is, doing things how she's going to do them, and her biggest struggle right now is just navigating the world. So that muscle function is extremely, extremely important. But then I look at someone like Dr. Matthew over there, and the same point, if we could improve brain function and that, that's also an incredible kind of thing to go on. So I don't know. I don't know. Yeah, they're both. Muscle is important, yes.
So Danny's severely impacted muscle. He had cortical visual impairment, or has cortical visual impairment. And I would take an intellectual disability, and it's supposedly pretty severe, but I take that with a grain of salt. I would do motor because as he's progressed up to 26, he was moving, doing he did everything but walk, etc. And what's been going on recently in the past, say, 5 years is the smooth muscles are going. So he never needed a feeding tube, and then all of a sudden, bam, at 12, he needed a feeding tube. He's got a neurogenic bladder now, and his coughing is getting worse and worse. So that's the part where he's getting sicker. We just kept him out of the hospital from a pneumonia because we have a new plan when he starts getting pneumonia. And that's what I see as the hardest part for him. I honestly don't, like, at this point, I don't care about the intellectual disability. He's very happy, he's very social, he loves people. And to me, it's what's gonna make it worse for him is he's gonna get septic from a pneumonia, he's gonna get septic from a UTI, and that's what's gonna be the end. Not to be, but that's what we think about, my husband and I, is like, and we do, like when I say I don't leave him, it's because we do have nursing now, but we don't have it all the time. And the bottom line is somebody's always with him. Like if it's not a nurse, it's me, it's my husband. And like we get up in the middle of the night and help him with his coughing. You know all the things. Everybody knows the things. But I'm just so like, he's the joy of our life. He really is. And I know he really is. He's the joy of our life. He brings a lot of joy. We have great times, and like I said, we made it to 26. We're very happy about that. And my husband's going to be really happy to hear, like, that all this stuff is going on. So I would say move.
I feel like we might be the odd ones out, but my son's only 3.5, so he has only so far progressed in terms of his motor abilities. He was delayed in everything, but now he's walking, he's like wanting to sort of run, he's very independent in terms of his mobility, and he's fully ambulatory. But he's nonverbal, and he has very low receptive language, and it's very challenging to do much with him in terms of like direction following and typical things that like a 3.5-year-old should be doing. And it's I think it's getting more challenging for him to not be able to communicate with us when he is unhappy, and he's expressing that with starting to show things like hitting his head, and we just talked to the neurologist about that too, which that can be typical also, but as he's getting older, it's getting more challenging, the intellectual things, I think more so because he is progressing at this point in terms of his mobility and muscle function. He's not obviously not typical, but once he started walking, I think it opened up a huge, it changed the trajectory for what everything was. He walked around a little before 3, so it's been over half a year, and he's, it's made a huge difference. So if I had to choose, I would love to be able to communicate with him and understand what he's going through and have him understand us.
Yeah, young parents or new parents of little kids is that the brain and the intellectual disability is the #1 priority. And so that's why in the last couple of rounds of grants, we were trying to fund research related to the brain consequences of the alpha-Dystroglycanopathies. And unfortunately, he couldn't make it today. He will come for the scientific meeting Monday and Tuesday, Dr. Kevin Wright. He works in Portland in the Volume Institute, and he is working with different animal models, mouse models, trying to understand the synapses, the connections between the neurons in the brain. And he just discovered and published a couple of papers of the importance of some of these glycosylation steps in the formation of the synapses and the maintenance of the synapses.
Where your cell is in the brain is written in stone, but how it talks to the other cells is not. And so that's the part that Kevin Wright is looking at.
Can I add? So that's also one of the things that I've always been interested in, because I'm a neuroscientist, so I work on the brain, and I had to, when I started working on dystrophinopathies, I had to learn everything about the muscle, and that's where you want to have the people that are hardcore developing the therapies for the muscle, and then also the people that are consistently thinking about the brain, because part of my lab also works on autism and intellectual disability. So I've been watching the drug development, the struggling drug development in that field very closely, because again, even if the brain, I have seen kids with brains that you wouldn't think they would be walking into the room, and then they walk into the room, right? So even if the brain, I'm sure you've seen a lot more, a lot more of those. And so even if there is an issue of how the brain is organized, there might be ways to push it to do a little bit better. It's never going to be 100%. And one more place for some types of dystrophinopathies is also the retina, because there is this progressive vision loss that can be very difficult, because if a kid cannot move and they cannot speak to you, and then they suddenly cannot see, and for some, you know,
So I've never seen progressive visual loss. We see developmental. Even there, I haven't seen it, but you see structural malformations of the eye. I haven't seen progressive visual loss that isn't correctable with glasses, even in adults with POMGNT1, but that's a very small N, small numbers.
So I talked to the POMGNT1 moms, and there's been like some issues with like retinal detachment, and then suddenly, you know, progressive. Yeah.
Myopia is different than progressive visual loss.
Right. Yeah. So we're also trying to figure out, we're in the fish, which is very similar as far as retinal structure, and Kevin Wright is also doing, also did beautiful work on the mouse retina to also try and see what happens there.
And we're looking at the human. So we published a very preliminary paper on looking at human retinal function with the concept that if we have a therapy, could that be a readout that we could use that is non-invasive, so it doesn't involve putting a needle in a human being, that is non-invasive, that could show an effect. So if the NIH ever gives us an answer, we may or may not get more information on the retinal function.
Yeah, I think it would be good if you have specific questions to pass around the mic and make, okay.
I'm going to just shoot my shot because I'm just so curious. I know Dr. Mathews, we were in touch with Carrie because we were initially planning to go out to Iowa for the study, and then we heard about this, and then here we are. But I'm curious about your overall experience with specific POMT1 patients, and if you'd be willing to share any of the progressions that you've seen. I know there's no crystal ball, and I know there's no way to know what the future looks like for our child, but we are sort of grasping onto anything that will give us some sort of insight, just because there is so little out there, just so we have some sort of expectations about his future. And I'm also curious, just to add on to that, with your experience with POMT1 patients, are most of them similar in what we described in terms of intellectual disability? Because, you know, we're in our second neuromuscular team, and our first team was kind of surprised at his nonverbal status and his intellectual delays, stating that they didn't think there was a connection, which is one of the reasons we sort of switched teams, because we were a little confused about that. And the second team said there absolutely is a connection, but I'm just, as you mentioned before, such a spectrum. So we're just trying to, I guess, like brace ourselves to see what the future might look like and any experience you might have with kids who are a little older.
So a couple things. First of all, we have a resident who is in theory writing up our POMT1, POMT2 experience combined. And I'm particularly interested in actually collecting data on the heart function, which I can't find very much written. I know it's an issue, but I don't know to what degree. And then we have another resident who with Dima and me is writing up sort of our entire dystroglycanopathy presenting before age 5. So we are going to have things out there in the literature that Matt can then, anyway. I think it just went off. Anyway, in the big picture, my general sense is that the motor function stays stable for at least periods of time, that my general, and this is going to sound weird, but my general sense is that the personalities are extremely strong. One might even say opinionated. Whether or not they tell you in words, or one or two words. In general, my experience has been that that group of kids has very strong personalities. You don't have any question about where you stand. Would that apply also to TRAPS C11? I think if I would tell you to expect continued slow improvement in what he is able to accomplish, with an endpoint that will be different than the norm. Those are sort of, I mean, it's not very specific, but there is a range, and
Kids typically regress in the same way that kids with like Duchenne's do.
so I would say most of my older POMT1, POMT2 patients are in wheelchairs. But there are also POMT2 patients who have a more limb girdle phenotype and a milder muscle phenotype. I don't, and Dima, you can chime in here too. I don't see necessarily, I wouldn't use Duchenne as an analogy. I would treat him as his self, and you will not fall off a cliff. If he's walking now, you will know if he's losing that ability. Can he right now arise from the floor? So if he starts to lose the ability to rise from the floor or climb, then you know that you're looking at something down several years down the line that's going to mean you're going to be looking at a wheelchair. I have patients with POMGNT1 who are in their 30s, have severe intellectual disability, walk just fine, except for the fact that they don't have good judgment or might have a seizure or something like that. Seizures are possible. Those are sort of, I don't think you're going to have any shocking. I mean, he could get hit by a car tomorrow and that would be shocking, but I don't think you're going to see a dramatic change overnight. I think you're going to have time to adapt your family's expectations in life.
Do you have experience with communication devices? Like, do you have a communication
I'm sure you are working with the speech therapists on that. And
He's in a specialized preschool. He's been there since he turned 3. He gets multiple therapies from a very comprehensive team. They have started working with communication devices. I think his intellectual delays are making it harder for him to comprehend how to quite use it. You know, for example, they had a PEX board and they had 3 items on it, and he would point to an item and then he would get upset because it wasn't actually what he wanted. So I think that it's a slow progression because I think his understanding is limited, but they're continuing to work on it. He has speech therapy 4 times a week, and we are seeing some improvements. He does have some approximations for words. He does have about 6 or 7 signs that he consistently and accurately uses. He is able to point to things and tell us things that he wants. He's very clear when he doesn't want something. But it's a slow progression.
And have you already seen any sign of seizures?
No,
No.
we have not. And we did have an MRI. I know you're talking about the brain. When he was admitted in the hospital, the time that we started our diagnostic journey for this, he did have an MRI, and there was nothing that came up in terms of his brain, structure, I guess. I know you were saying, like, once there is a brain thing, it can't really be changed. So I don't know if that's, giving us some hope or what, but there was nothing remarkable that came back in the MRI.
Well, we also don't know if the doctors didn't really know what they were looking for.
Right, it was like
you can have, this is true for LAMA2 too, you can have microscopic abnormalities of the cells going where they're supposed to go that you can't see on an MRI. If he may never have a seizure. If he had a seizure, it wouldn't be shocking and oh my gosh, we have to look for some incredibly esoteric problem.
This is something that our new team at CHOP talked about, was the seizures, which our former team never mentioned, which is something that we're now on the lookout for.
And
Diagnosed with limb-girdle, but he didn't get diagnosed till he was 13. He didn't get the gene till he was 17. And I used to do Dr. Google and put all his symptoms in, and guess what came up? Fugatin, all the time. And I would always say, because he had his diagnosis with ataxic cerebral palsy. And so the seizure started when he was like 4. But I think he was seasoned before then.
It was something that came up as something to watch for in some of the literature. However, we haven't seen any. One just kind of, I don't know if it's funny or not, but she was at early intervention UPK, and there was a game that they were playing where they had the freeze on the TV, like they're talking, and we had been playing freeze with her, and I got a phone call that my daughter was having a seizure because she just froze during the game and did not come out of it for like 10 minutes. And it was terrifying at the moment, but it's kind of a funny little thing to, and that's her personality. It's something that she does, but it was not a seizure. It was something that, yeah, it was immediately, I came down there and, my wife is like, did you try telling her to unfreeze? And it worked. So.
Yeah.
Listen, PEC sucks, okay? I'm a special ed teacher. Danny doesn't talk. Of course, he can see that we didn't know because he had cortical visual stuff. Voice output. Have they started with voice output at all?
I'm not familiar with it.
Okay, so voice output is basically, like he has actually, he's 26. We know what the kid wants and doesn't want. Like, he's got all these like idiosyncratic ways of letting us know what he wants and what he doesn't want. But he does have a tablet, and unfortunately for him, because his intellectual disability is enough that, but he's supposed to like hit something and then it goes to a board, and then it goes to something else, and it was just like too many steps for him. But voice output, it's so much easier than the PECs, and then they can put sentences in, like, I want a, and then ball, I want a, he can have a whole thing. They can tell jokes on it. So tell them, and you're gonna, and I'm gonna tell you right now, as a special ed teacher, you tell them what you want. Yeah. And if they don't let you, then you get an advocate. Get an advocate. I didn't even know that.
Get an advocate in the school.
Yeah, just someone who knows the laws and even just knows the key words, because a lot of times in these special ed meetings, if as a parent you say something a certain way and use certain language, the school has to respond, has to at least acknowledge what's going on. And I don't say don't believe what they tell you, but be persistent in what you want. And if you want something, 90% of the time you'll be able to get it as a parent pretty well. The schools are underfunded, and they're doing what they can to get by. And if you need something and that is the best thing for your child, they have to make it happen once it's written down. So they're going to try to do everything they can to, oh, let's try this, let's try something else. But as if this is the best thing for your child and you get it put in that document, they have to figure it out.
And you can be difficult and be nice. Because I'll tell you, as the special ed teacher, when we get lawyers, I've been in plenty of meetings with lawyers and advocates, I don't really care. Because I have my own, I have my own experience with it. So it doesn't, like, if you want to bring a lawyer, good. I'm glad, I'm happy. Because if I'm doing something wrong, I'll change it. However, it scares administrators so much when you bring an advocate and you talk about lawyers. They're like up the special ed teacher's butt. They're like, hey, come on, you got to, oh, so-and-so's coming, you know what she's like. And be that person. Because our kids are so rare. So we have to.
Okay.
This one's, I guess, I have two. One kind of a specific question. There's only really like 6 studies that I've ever been able to find on INPP5K. And this one is about zebrafish specifically. So I have the person in here who will know what I'm talking about. There is now I can't even find it, a study talking about L-serine. Yeah. Now, L-serine, is that a completely, if I do the over-the-counter thing, is it just I'm going to digest it and turn it into a different protein and it's a complete waste of my time and money? Or it can't hurt and this isn't going to get funded enough in Nova's lifetime to be helpful to find out. So...
So the reason why we made the knockout, which is a fish where there is a mutation that breaks the gene, is that study had used the knockdown, which means they reduced it to a certain amount. There is a lot of issues with the MPP5K in fish, which is related to fish. Most of the fish genome, every single fish in the sea is duplicated. So IMPP5K exists in like 2 separate copies, which is different from the humans. So when you have to study it in fish, it's complicated. And so they had to do this like double approach. The particular approach that they used is known to sometimes just cause physical issues with the fish that have nothing to do with the gene, but have to do with the approach. So I don't know. It looked interesting when it came out. It was like a few years, so it was before. And so we did a lot of work trying to figure out which one of the two we wanted to break, and that made the mutant. To tell you the truth, the knockout fish dies within a week, so which is very different from, or no, it's not, because the humans are all changes in the protein, so there is really no human that does not have IMPP5K. And so the next step would be to start putting in all of those changes into the fish and see what happens, but I don't know. Would taking L-serine do something? Because you have to ask a doctor. You don't have to ask.
I don't. Well, I was going to defer to you. Is biologically, it doesn't make sense to me.
Make sense.
I was going to ask if there was something in the pathway that I was unfamiliar with.
I mean, biologically, it doesn't make sense, and so I wasn't sure what that was about, and I wasn't sure whether it was something due to what they were doing to the fish that would suddenly make it better.
There's no way everyone who is much smarter than me and missed this and didn't tell me just immediately start taking these. So, okay.
There's a different neuromuscular genetic condition that L-serine works for, but it's a totally different and unrelated and not anywhere in this pathway. So I have no idea. I don't know. In general, I know we had this is research, not care, but what the heck. And I know we had a nutrition session this morning, and they didn't come out, and I don't think explicitly stated this, but if you do something like L-serine, you don't know actually what's in that bottle. So as you probably know, when independent pharmacies go and review what's in the bottle, it's about 50-50 whether what's on the label comes close to matching what they say it is. So in general, as a pediatrician, I do not recommend giving things that are not FDA regulated without having a pretty compelling reason. If your vitamin D level is 5, and then you definitely need to take vitamin D3. But I'm not a fan of supplements for all those reasons.
Which is why I'm sitting on there for 2 years. Never done anything or pursued it further. I had the person in the room who would know the answer to that question. The second kind of thing, and I know we're going to have an advocacy session later, but I'm meeting with Senator Schumer next week, and I'm going through like Rare Across America, like that kind of organization for specifically rare diseases as a way to group people together. And I know that we can also group together through the CMD kind of as a way to do it. Would it be helpful to mention Rutgers and Wellstone by name during these kind of things? Is that like, wouldn't hurt? So I guess I'm kind of curious as to where the most effective, and I wish you probably also wish you knew the answer to that.
Schumer is my senator. I've been calling him on like a weekly basis, mostly to yell at him. So, I mean, the point right now, so we're going through, you're going next week because it's appropriations season, right? There is a request to slash the NIH budget by 40%. Last week, the Appropriations Committee at the Senate said no, and they, bipartisan, but basically they said no. I think it was 400 million, or like it was a small increase, but it was an increase on the budget, right? So right now, really the message is protect NIH budget, increase NIH budget, make sure that because any cut would disproportionately affect rare diseases, just by virtue of the fact that they'll prefer when you pick for, I'm reviewing grants right now because I have to turn in the reviews for the NIH, right? You have now significance and how important something is the top criteria that they're going after. And so when you're competing, I was competing for the dystrophinopathy therapeutics in a review group that had all diseases, right? I'm like, if there's a good gene therapy grant in any other part of the body, they're gonna win over me, right? They're gonna fix it. I'm just asking, because there is absolutely nothing. I'm asking to be able to find something that's gonna make the kids better. But right now, the hit rate is less than 10%. If there's one grant in, another disease that is easier to tackle, they're gonna win. But the problem is that the diseases that are difficult to tackle are going to lose. And so the advocacy really at this point is very, very, very important. I mean, and I'm sure.
And I think that I think advocating for biomedical research, I'm a clinician, but advocating for biomedical research, because you don't know necessarily, if the science is good, the hypothesis is good, it's well developed, it's logically supported, you don't know what science is going to lead to the thing that makes a difference for a rare disease or a common disease. So my argument would be, and if all we're doing is focusing on the end stage, I'm pointing at the diagram, but if all that basic research that seemed pie in the sky 20 years ago, I mean, when we started doing this, Fukutin or FKRP was like the only really known gene of the dystroglycanopathies. We knew nothing about it. If basic scientists doing insignificant work hadn't worked out the pathway for developing this glycosyl chain, we wouldn't be doing clinical trials today in FKRP and potentially laying the groundwork for future dystroglycanopathies. You have to do work that doesn't, and if the focus right now is it going to treat 1 million people with hypertension tomorrow, you don't know where your research dollar is going to end up. That's.
Yeah, it's time, but I, I just want to add that fund research is NIH, for example. So, and can mention the case of Kevin Wright, that we gave him a grant for 50,000 and for something that was very new, nobody did it before. He did it, got good results, and then he got MDA grant that was 300,000, and then now he has an R1 from NIH that is $1 million and something for 5 years. But it seed money anywhere, so it's very important. Well, thank you, everyone. Thank you.